Welcome, everyone. Thank you for joining us for today's webinar, Do You Have the Gut on a Chip? Presented by Dr. Elena Naumovska, Scientist of Application Development, Mimetas, the Organ on a Chip company. My name is Xavier Gutierrez, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Now, before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. And if you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Dr. Naumovska. I will now turn the presentation over to her.
So hello and good day, everyone, and welcome. First, I would like to thank the organizers for inviting me to present this webinar on growing perfused epithelial tubules in a parallel microfluidic platform. I would start my first slide with the mission of Mimetas, and that is to provide human and tissue organ models for the 21st century therapeutics. And with this, we are hoping that we will lead to a better medicines and personalized therapies for the future. Today, I'm going to talk about our intestinal models, and I would like to start with addressing some of the limitations of current models. Then, I will introduce you to the OrganoPlates, which are the platform that we use to create these models. Then I will go in deep on how do we develop intestinal tubules on a chip, the available assays that can be used with these models, and I will show you a little bit of the results that we've gotten with a compound screen. I will also talk on how you can add complexity in the organoplates. And in the end, I will explain it to you how you can also start using the platform in your lab. So, the current in vivo and in vitro gut models are lacking in certain aspects. Preclinical research remains an important tool for drug discovery and validation of new therapeutics. And animal models so far have played an important role. However, in most of the time, they have limited use in predicting the clinical relevance. One reason can be that animal models metabolize drugs and are affected by disease differently than humans. They cannot accurately recapitulate the human condition simply because they're not people. In 2D conventional models, there is a problem because cells do not behave the same in 2D as in 3D, and most of the models of the intestine lack the important transporters, which are fundamental for studies. Moreover, when cells are seated on artificial membranes, they do not recapitulate the normal physiology. These are artificial membranes that produce a physical barrier, and there is a lack of cell-to-cell and cell-to-ECM interactions, which normally happen in proper microenvironments. Another thing to consider is that 2D cultures need a long time to differentiate and mature, and for some can take up to 21 days of constant testing to be ready for assays. And this is mostly done in a low throughput format, meaning that there are only 6 to 24 replicates at the same time. This is also connected to high reagent consumption. Moreover, in static 2D cultures, there is lack of flow, which is known that it's needed for proper differentiation. So what we think here at Mimetas is the right way to go is not to use 2D cultures or animal models, but rather try to recreate the in vivo situation in the human body as good as we can. The way to do so is to provide a 3D environment to our cell cultures as close as possible to the in vivo situation. Because it is widely known that ECM composition can serve many functions, such as providing support, segregating tissues from one another, and regulating intracellular communication. Another thing that we consider is an implying flow, because flow can emulate physiological microenvironments experienced in vivo. And we also pay close attention to providing the right cell-to-cell communication between the cells when we build complex co-culture models. To set this up, we use the OrganoPlate, which is a 3D culture device that is developed here at Mimetas. And as you can see by the images, it looks like a standard 384 well plate. And that is because this is a standard 384 well plate on the top part, meaning that it's fully compatible with all the standard laboratory equipment. However, when we turn the plate, we can see that there is a special part about the device, which is the bottom compartment. This is made of a sandwich of two glass plates, which are microscope grade. In between these glass plates, there are walls that form the microfluidic channels. Moreover, there are holes drilled in the top glass plate that allows access to the microfluidic channels underneath. The layout is designed here at Mimetas, and we are also flexible in producing different designs. And the one that you're looking at is the 3-lane format. This means that there are 3 microfluidic channels in this plate, and exactly 40 of them. So 40 small experiments at the same time. We also have a 2-lane design, which has 96 chips. On this image, you can see the 2-lane format, meaning there are 2 microfluidic channels, and the wall of the microfluidics, which is in blue, embedded between the 2 glass plates. What you can see in this image are 3 2-lane chips here. To add the extracellular matrix, we use a specialized technology to fill this microfluidic channel, which is called the phase guide. And in the image, it's represented in green line. It is basically a small ridge between 2 microfluidic channels, approximately 1/4 of the height of the microfluidic channel. And it functions as a pressure barrier. The extracellular matrix in this image is represented in red. And as we add the red ECM in the first chip, the ECM enters by capillary force, and it's pulled into the channel. The phase guide prevents this overflowing to the next channel because of the surface tension of the liquid. This can be done by any extracellular matrix that is liquid at lower temperatures and gets solid at higher, meaning that you can create a layer of ECM with or without cells in one of the compartments while keeping the top compartment empty. Leaving the top compartment free is extremely important when we want to make intestinal tubules. On the left side, you can see the OrganoPlate and an art representation of the 3-lane chip. And underneath, there is a schematic representation of the procedure for tubule seeding, and how does it look in the different planes. We have the ECM in red, and the cells are represented in dark blue. In the first step, we add the extracellular matrix, which enters the channel and runs into the phase guide, which prevents it flowing into the other compartments. Once the ECM is gelated, we then add the cells resuspended in medium in the top channel. Cells are left to attach to the ECM by gravity, after which we start applying flow. What we have seen so far in all of our epithelial and endothelial cultures tested is that we don't just form a boundary structures against the ECM, but they also form a tubular structure in that channel. On the right, you can see also the intrinsic impressions of the tubules grown in a 2-lane format on the top and a 3-lane format in the bottom lane. To create flow, we make use of a rocker platform by simply placing the plate, and there is no tubing or pumps involved. The fluid flows through the tissues growing in the OrganoPlate, is driven by gravity leveling. By simply lifting one of the sides higher than the other on defined intervals. We are recreating a volume difference, and we create a bidirectional flow in this manner through the media. To make it go continuously, the plates are simply placed on the perfusion rocker inside an incubator. This perfusion rocker features a time program designed for the OrganoPlate, but for evaluation purposes, a standard continuous rocker platform can be also used. And this is how a 3D reconstruction of one of these tubules look like. In the bottom part, we have the perfusion lane, which there is a lumen. We have a representation of the phase guide and the gel. As you can see, since these are tubular structures, they're also accessible from both the basal and the apical side. The great thing of the OrganoPlate is that we don't have to limit ourselves only to tubular structures. And in this overview, you can see part of the models that we have developed using the OrganoPlate. In the left, you have the tubular structures like models of the intestine, vasculature, and kidney. However, we can also see the cells into the extracellular matrix and have models of the liver, brain, and to grow organoids in gel. And if we combine these two features, we can also have a complex co-cultures, like the blood-brain barrier model that we have, the kidney clearance model, and endothelium parasite model. However, in my talk, I will only focus on the intestinal models. So why are intestinal models important? Oral drug delivery is considered the preferred route of administration because it's user-friendly, it reduces risk of infection, and reduces pain and suffering. Moreover, transport of drugs through the intestinal membrane is a complex and dynamic process, and this is important in the candidate selection in the drug development processes. Another thing to consider is that there is really high difficulty in controlling parameters in vivo models. Therefore, here at Mimetas, we developed a simple 3D intestinal model based on the CACO2 cells. These are human intestinal colorectal adenocarcinoma cell lines, but they have a really interesting feature that once they reach confluency, they start to differentiate. And when differentiated, they resemble enterocytes of the small intestine. On the right side, you can see a phase contrast images of a tubule formations at day 1 and 4. On day 0, the cells are seeded against the extracellular matrix, and they start colonizing the glass walls to form a confluent tube. Here, perfusion plays a crucial role for tube formation. What we found in our cultures is tubules are formed in 3 days, and an optimal barrier is found at day 4. But we can also see that not only did they form a tubular structures, these cultures differentiate in 4 days, which is a surprising fact, keeping in mind that standard 2D cultures of these cells need 17 to 21 days to do so. And in this image, I'm showing you the stainings for tight junction, which is ZO1 in red. We also have the microvilli markers in green, and the nuclei are stained in blue. But these cultures also stain for different transporters like PGP, GLUT2, MRP2, ERP1, and ERP2, just to name a few. As I mentioned, that the optimal barrier function forms on day 4. To assess this, we use a real-time barrier integrity assay, in which we perfuse a fluorescent probe with defined size, which is added in the culture media through the lumen of the tubule. When we have a leak-tight tubule or fully intact barrier, the fluorescent probes are retained in the lumen. But when there is a damage to the barrier, like a toxicant or a drug, then the fluorescent probe leaks into the gel to the basal site, yielding a higher signal in the ECM. We measure all this by a time-lapse fluorescent microscopy, and we use a quantification method where the fluorescence level measured in the gel region is normalized to the fluorescence level in the tubule. In the end, we get this kind of graphs where we can assess the leak tightness of the tubules. And this is extremely important when we are assessing drug exposures. In this image, you can see the effect of a drug concentration over time. With increasing drug concentration, we can see an effect on the barrier integrity, even after 9 hours of exposure, while the lower concentration of the drug don't affect the leak tightness of the tubule even after 24 hours. This type of assay was useful when we were assessing the barrier integrity of a 4-day-old CACO2 tubules for a Nature Communication paper. Here we expose the tubules to a various concentration of staurosporin, which is a known inducer of apoptosis, over 124 hours. Fluorescence levels were measured for both 4.4 kilodaltons and 150 kilodalton dextrans, in which we determined the liquid scores, the survival curves, and the EC50 time curves, which provides an insight into both concentration and exposure time response. This study was comprised of 357 gut tubules with over 20,000 data points, making it the largest published organ-on-a-chip dataset so far. But not only that we can model healthy gut, we can also model diseased one. In the next slides, I will show you the application of the robust 3D gut-on-a-chip model for investigating some of hallmarks of inflammatory bowel disease. This is a term for 2 conditions known as Crohn's disease and ulcerative colitis, and they are characterized by chronic and extensive inflammation of the gastrointestinal tract. In the OrganoPlate, using an immune-relevant trigger, we can recapitulate key aspects of IBD, like morphological changes, which you can see from the phase contrast images once we apply the inflammatory trigger, and not observed in the control tubules. Another key aspect of IBD is the loss of barrier function of the intestinal epithelium. In the top image, we can clearly see this after applying the inflammatory stimuli. And as we can examine further, we can also observe an activation of the intestinal epithelium by increasing the expression of pro-inflammatory molecules and decreasing the expression of differentiation markers once the trigger is applied. So this model makes a very valuable tool for high throughput screens, both involving target knockdowns, for example, an adenoviral screen, which can be done directly in the organoplate, or with IBD-specific compounds. In the previous slide, I showed you the barrier integrity assay and the use of it, but we can also use a more precise way to assess the integrity and permeability of monolayers, and that is to use the transendothelial electrical resistance, or TER. This method is based on measuring ohmic resistance, or measuring the impedance across a wide spectrum of frequency. For this, we have a new device that can measure all of the tubules in one go in the organoplate at the same time, and it's called the OrganoTear. The TER measurements here are extremely valuable when the BI assays cannot detect the difference in the barrier integrity, especially with cultures that develop a stronger barrier integrity, like intestinal tubules. In this figure, I'm just showing you the results that we got with one of the compounds tested in reversal of the IBD phenotype. And we can not have only monocultures, we can also have co-cultures. And in this slide, I'm showing you the co-culture developed because we also wanted to have a mucus production. Here we have a monoculture of CACO2, a monoculture of HD29, which don't have barrier function, and a co-culture of CACO2 and HD29. They do grow and form tubules. They have a nice barrier structure, and they also stain nicely for most of the transporters. And in these cultures, we do have a mucus production. And this is just a 3D reconstruction of the same cultures. Here they're stained for the microvilli and MUC2, which is also another mucus. But not only co-cultures. In the organoplate, we have the option to have really complex models. And in this slide, you can see the co-culture on the top, the mucus producing one, named as gut tubules. In the ECM, we also have incorporated cells, intestinal myofibroblasts. And in the bottom channel, we have a tubule of endothelial cells perfused with immune cells. These cultures are stable for more than 15 days, and they can be used after day 7. And you can see also the stainings for their markers in the bottom part. So what cells you can use in the organoplate? Basically, any immortalized cell lines, whether it's human or animal. And I just gave you some of the examples, like CACO2, HD29, or immune cells, here, THP1 and MUTZ3. You can also use primary cells, like intestinal epithelium or isolated immune cells. We can also use organics or embryonic and induced pluripotent stem cells. And the platform is highly compatible with all the high-content imaging systems, but also with normal imaging systems like phase contrast microscopy, fluorescent, and confocal, and with all of the standard laboratory equipment, like absorbents and plate readers. And is compatible with automation. And these are some of the assays that can be used in the OrganoPlate. And since we can also sample and have access to both the basal and apical side of these tubules, we can perform an off-plate assays. So the OrganoPlate can be brought to your lab. It has the unprecedented image capacity, so you can do live imaging, and it is commercially available and it can be ordered from our website. So in the end, I want to thank our team. This cannot be done without their help, and also many of our collaborators. And again, these are just some of the models that can be done in the OrganoPlate. So thank you very much, and I'm looking forward for your questions.
Thank you, Dr. Naumovska, for that informative presentation. I would also like to thank LabRoots for making today's educational webcast possible. Now, before we go, I want to let everyone know that this webcast will be available for on-demand viewing through May of 2019. And as a final reminder, our speaker will follow up with any questions you've submitted via email. And that's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.